Hello, my name is Krithi Irmady. I'm a movement disorders neurologist and physician scientist at Rockefeller University. The goal of my research project is to investigate RNA regulation in Parkinson's disease and levodopa-induced dyskinesia. My project specifically focuses on understanding the mechanisms that regulate the function of a brain region called the striatum. The striatum is critical for both planning and controlling movement, whether it's eating, exercising, or running. When it doesn't function properly, the control of movement breaks down. In Parkinson's disease, this dysfunction leads to many symptoms of the disease, such as slowness in movement. Understanding why the striatum stops working correctly in this disease is at the heart of my research. Parkinson's disease occurs because dopamine-producing neurons in a different region of the brain, known as substantia nigra, die. These neurons normally supply dopamine to the striatum, which is essential for the striatum to work properly. We can replace this lost dopamine with medications like levodopa, which help improve movement to some extent. However, these medications, especially at higher doses and when given over a long time, can cause involuntary, erratic, abnormal movements known as levodopa-induced dyskinesia, which can interfere with daily life and limit the effective use of the available treatment options like levodopa. My project aims to understand what goes wrong in the striatum, both in Parkinson's disease and in levodopa-induced dyskinesia. To investigate this, we study brains donated by individuals with Parkinson's disease and compare it to brains from donors without Parkinson's disease, who we call controls. We also compare brains of Parkinson's disease patients with and without levodopa-induced dyskinesia to see what makes them different. We focus on RNA, the molecules that encode genetic information from DNA to make proteins, which are the building blocks of every cell. We study two things. One, what are the RNA changes that occur in the brains of patients with Parkinson's disease and levodopa-induced dyskinesia? And two, how do factors known as RNA binding proteins interact with RNAs to affect their normal function? Why is it important to study RNA in Parkinson's disease? It is important because RNA doesn't just carry genetic instructions from DNA. It also controls how much, where, when, and what kind of proteins are made in cells. If RNA function is disrupted, neurons in the striatum cannot communicate normally with each other, which is crucial for normal brain function. The function of RNAs can be disrupted because of how RNA binding proteins interact with RNAs in the context of a disease. Our research has already shown that changes in RNA and RNA binding proteins can lead to abnormal striatal function in animal models of the disease. Now, we're extending this work to human brain tissue to uncover what drives the clinical symptoms of Parkinson's disease and levodopa-induced dyskinesia at the molecular level. By understanding these changes, we hope to identify new ways to improve treatments and reduce the burden of both Parkinson's disease and levodopa-induced dyskinesia. Finally, I want to thank the APDA and all donors for their support, many of whom may have been personally affected by Parkinson's disease. Your support is critical to advancing research and finding new ways to tackle Parkinson's disease. Thank you so much for making this work possible.